I'm very interested in patients with myeloma and what we can do to learn about the multiple myeloma from blood biopsy. And there's two modalities of blood biopsy that we are interested in, and those are circulating multiple myeloma cells and cell-free DNA. And we have established various genomic technologies that allows us to interrogate cell-free DNA and circulating myeloma cells. And we started with circulating myeloma cells and did deep genome sequencing, and we found that we can actually reproduce the exome, the genome, and also the transcriptome from single cells that we can isolate just from a vial of blood. And similarly, we isolated cell-free DNA from a vial of blood, and we found that we could also do whole genome sequencing and whole exome sequencing from a vial of blood. And the purpose of that is really threefold. There are three potential advantages. And the first one is very obvious because it's much more convenient for a patient with multiple myeloma to just have a vial of blood drawn than to undergo a bone marrow biopsy. And the second advantage is that you may actually be able to get additional information, because when you do a bone marrow biopsy, there's always a sampling bias, and you pretty much sample just one particular site in the body. The hope is that with blood biopsy, you get a much more representative picture of the myeloma in the entire body. And so we have been developing those technologies to really see if we can reproduce a bone marrow biopsy and in some cases even forgo bone marrow biopsy, that's the hope anyway, but also to get potentially more information that we would from a bone marrow biopsy alone. Some of the limitation for cell-free DNA is really the amount of myeloma-derived cell-free DNA that is present in the bloodstream. And there needs to be a pre-selection of patients, which can be done actually with very cost-effective tests to figure out beforehand which patients would be, for which patients it would be feasible to use cell-free DNA instead of the bone marrow, to just do the genomics. But I think in terms of technology, we are not that far away from implementing this in clinical routine. The challenge for circulating cells is that it's much more complicated and it's much harder to standardize. So those are technological concerns. I think one of the concerns that people have also raised is the fact that when you isolate myeloma cells or also cell-free DNA from circulation, you are missing the microenvironment. So particular things that you're looking at, for example, when you're looking at the transcriptome with RNA sequencing, you're missing the microenvironment. So there might be relevant clinical, biological, and also therapeutic differences between a myeloma cell that you have isolated from the bone marrow compared to a myeloma cell that you have isolated from the peripheral circulation.